If you ever walk along Baltimore's Inner Harbor or any other urban waterfront, you'll see plastic trash everywhere. It's not hard to see how garbage like takeout containers, plastic bottles, and grocery bags affect our waterways. What we may not see, though, is how this trash breaks up into smaller and smaller pieces and forms what is known as microplastics. Microplastics are plastic particles that are smaller than 5 millimeters. That's about as big as an eraser on a pencil. They can come from many different sources, and they can be classified as primary or secondary. Primary microplastics are those that were made to be that small, such as microbeads or microfibers. Secondary microplastics are those that are formed when big pieces of plastic debris break up as a result of physical stress like waves or sunlight. These particles are present in most bodies of water around the world, and their true impact on ecosystems and their organisms is only starting to be understood. I am a microbial ecologist. This means that I study how, where, and why microorganisms like bacteria and microalgae live. For my doctoral research, I am looking at how bacteria interact with microplastics in estuaries. So far, we know that bacteria can attach to plastics and form complex biofilms, but we do not yet understand how these biofilms form or what they can do. I believe that the type of plastic these fragments are made of can significantly impact the type of bacteria that attach to their surfaces and how they interact with each other and their environment. Plastics that are dense and tend to sink, like polypropylene or PET, will interact with different bacteria than those that are less dense and tend to float at the water's surface, like polystyrene or styrofoam. All of these plastics make up a great portion of the debris polluting estuaries like our very own Chesapeake Bay. In my experiments, I take microplastic beads and I put them in containers that I later deploy in Baltimore's harbor to be colonized by bacteria and other organisms. After some time, I take them back out and I look at the communities of microbes attached to them by analyzing the DNA fingerprints in their biofilms. By understanding what these biofilms look like, how their diversities differ, and how they are formed through time, we can focus on the environmental impact that they can have. Bacteria are responsible for the global cycling of the elements and can even impact our climate. If plastic particles covered in biofilms are present in the water in high enough concentration, they can change how the entire microbial community in the estuary grows and develops. Microplastics can also be home to harmful microbes that can be toxic to animals and invasive to ecosystems. Preventing and managing plastic pollution is a huge priority. Big pieces of debris have been the focus of these efforts for decades, and we're now just starting to understand the extent of the problem of smaller plastic fragments. When you're walking along a waterfront and you see a bunch of garbage floating, imagine how many microplastics are there but you cannot see. We're talking billions. Through my research, I'm shedding light on how they affect the environment and working on discovering this problem's true size.